Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Modernizing Viral Clearance Study Strategy, Regulatory Updates, and Industry Best Practices. I'm Jokubas Liekauskas, an editor at BioInsights, and joining me today is Kathleen Kenny and Audrey Chang, who will explore best practices for viral clearance studies, insights on ICH Q5A R2 updates, and expert guidance on designing phase-appropriate studies to ensure product and patient safety. After representation, we'll have a live Q&A session, so feel free to pose your questions to our speakers using the Ask a Question box at the bottom of your screen, and we'll try to get to them during the session. Now, I would like to introduce our presenters. Dr. Audrey Chang, with over 28 years of experience in biological product testing and laboratory management, leverages her expertise from the FDA, BioReliance, and Bigene Biosciences to provide technical solutions for biologics and advanced therapies at WuXi Advanced Therapies. Kathleen Kenny, Director of the Viral Clearance Team at WuXi Advanced Therapies, brings over 13 years of experience, progressing from lab technician to leadership with extensive expertise in study design and execution. So without further ado, I'll hand over to Audrey to kick off today's presentation.
Thank you, Jakubas, for that kind introduction. Hello, everybody. On behalf of Kathleen and I, we're happy to be here to give our presentation on viral clearance. I'm Audrey. I will start it off, lay the groundwork, a little bit overview, and then I will turn it over to Kathleen for the good stuff. Next slide, please. So WuXi AppTech is actually made up of 4 units that offer end-to-end contract testing, manufacturing, discovery, and development. Both Kathleen and I are with WuXi ATU, Advanced Therapy Units, located on the left, where we focus on manufacturing of advanced therapies and as well as testing at our Philadelphia, Pennsylvania campus. Can I have the next slide, please? So at ATU, we're very fond of saying enabling DNA to BLA because our services actually encompass the entire drug development process from early preclinical all the way to commercial. And we have 3 main platforms. We're known for our viral vector manufacturing, as well as cell therapies. Perhaps we're best known for our cell therapies, and for our GMP testing that supports not only our manufacturing clients, but also supports many external biological manufacturing, ranging from the traditional biological products such as monoclonal antibodies or recombinant vaccines to some of these more advanced modalities. And you'll see at the bottom of the slides the number that's associated with this Philadelphia campus. We've been completing over 59 projects. We employ over 900 people that work at that site and have produced over 2,500 CGMP release tests for products. Can I have the next slide, please? So pictured here are the 4 buildings that make up our Philadelphia, Pennsylvania campus. 3 of them are totally dedicated to manufacturing, from process development, early phase and early clinical, all the way to fully late phase, as well as commercial products. But if I could focus your attention to the building that's on the bottom left-hand corner of this slide, that is our testing facility. And this testing facility, all 140,000 square feet of it, was designed and built to do one thing, and that is GMP release testing. Housed here, we have all of the over 700 active assays, encompassing all of the scientific platforms. We have, microbiology, molecular biology, cell-based assays, analytical, and of course, cell-based microvirology, molecular, and analytical. Those are the 5 platforms we have, and they're all used to do the lot release testing of biologics testing for the materials that we have. It's nice that we're all co-located on one campus. It obviously it simplifies sample transfer, but we find that co-locating testing with the manufacturing allows what I call an ecosystem where our scientific and quality experts can collaborate and interact each day. Can I have the next slide, please? So just the guiding principles of quality control testing, what we do here, um, it's mandated by regulatory agencies worldwide, but they all follow the same common principles for quality control tests. It assures product acceptability, that your product specs must be scientifically based, and it demonstrates that you have overall control strategy of your manufacturing process the entire time. And these tests fall into different answers to different questions, the product that you made, which I've illustrated on the right hand side of this slide. It shows that, identity. Have you made the right product, and is it free of unwanted both process and product byproducts, potency and quantity, how much have you made and how well does it work, the quality attributes that you might have with your product, and safety, which is the focus of today's talk. Safety. Is your product free of unwanted mycoplasma, viruses, adventitious agents? And so what we're going to focus on today is viral safety. And the next slide, please. So what I've shown here is the types of studies that you have to do, and they fall into three buckets. Taking a step back, modern biologics actually has a very strong viral safety records, and this is really due to the CGMP manufacturing practices that we put in place. And they fall into these three categories, often referred to as a biosafety testing triangle. This approach actually works very well for all biologics, whether you're making a monoclonal antibody or a complex allogenic cell therapy product. So the first part of this triangle, or the first pillar, is it's called characterization. You have to select materials and test anything that goes into the process for the raw materials, the cell banks, and media. And the second part of this triangle is your lot release testing, or referred to as batch release testing, where you have to test the product at prescribed points in your manufacturing process with a prescribed testing panel. And the third one's clearance testing. Now, clearance is a little bit different. This is a study where you're actually assessing the capacity of your production process to either remove or inactivate, render it non-infectious, but actually testing the process to show that it can purify away any unwanted contamination. So a clearance is a little bit different. So characterization and batch release testing, those focused on direct testing, meaning that you take the sample and then you test it. And they may have its limitations. So for instance, you may be only looking for known viruses. You may potentially miss new variants or novel viruses that might enter the process. Finally, the direct testing is based on assays that have a limit of detection, so maybe very low levels will not be detected. So this is why the third piece of this triangle, clearance, is very important in balancing and assessing the viral safety. Can I get the next slide, please? So very quickly, I mean, the basic, simplified what viral clearance study, it actually provides data. You actually provide evidence that your manufacturing process will clear, which is a fancy way of saying remove, or inactivate, which would mean render it non-infectious, your product. So you actually do a study where you're going to put in or spike in amount of virus, run it through the process, and then see how much is either removed or cleared by that process. You could either remove it by a column, or perhaps you could render it non-infectious by chemical inactivation. And so this is a very, again, it's not direct testing, but it actually shows through data that your process has the ability to remove viruses, or if a contamination did occur. And so this is what we call the manufacturing process specific study, because you have to select a step, you have to design and validate a scale down model, and of course determine what types of viruses you're going to spike. And Kathleen will go into those details a little bit more in her part. Can I have the next slide, please? So, our viral clearance setups and modules, we've learned a lot through what I call the monoclonal antibody age. We've come to a point where we really truly understand not only the monoclonal antibody, but also the manufacturing process. But cell and gene therapy, as we all know, is quite different. It's a different from a monoclonal antibody. It's a the product itself can also be considered a living product. It can almost be virus-like. So the manufacturing process will be quite different, right? You may not have a step that could remove or inactivate uh, potential virus contamination while without compromising the actual product itself. So if you're not able to perform clearance studies, I sort of illustrated that there by putting a little red X if you have a cell therapy product, for instance, you're going to have to say, well, how do I assess the biosafety status of my product? And what you're going to have to do is rely more on the other two parts of this triangle. You have to do more extensive characterization, and you perhaps to do additional batch release testing, because again, you have to assess the biosafety of your product. So what I'm trying to say here is that, biosafety assessment is not a check box type of an exercise. It really is assessment of based on the scientific knowledge of your manufacturing process, and also doing a thorough risk assessment of the entire process to understand what potential contamination might be coming in. If I go to my last slide before I turn it over to Kathleen. So the regulators actually understand that the cell and gene therapy modalities are bringing a new complexity when it comes to virus safety. And they issued a long-awaited guidance at Q5A, the revision 2, where it actually upstates the thinking and represents the key understanding from a regulatory point of view. It retains the same key principles from the original one, the biosafety triangle that I presented earlier, but it does acknowledge the unique nature of cell and gene therapy products. It actually expands viral clearance scope to viral vectors that perhaps are amendable to clearance studies, such as AAV. It actually calls out that if your manufacturing process includes the use of a helper virus, you have to demonstrate clearance of that. And again, it also reiterates that viral clearance studies should be designed phase appropriate, meaning, the design of an early phase study may be less complex, less cumbersome, versus a late stage study where you have to do more studies, more steps, and more different model viruses. So unlike a MAB where you kind of do a risk assessment with a log reduction value, that's a fancy way of saying how much virus I have to clear. With cell and gene therapy, you have to determine it through a risk assessment, understanding what my methods, if I used a helper virus, or did I just have triple transfection? If I have a wild type capsid, do I understand its susceptibility to inactivation conditions? If I have a novel capsid, does that change? Again, using your scientific knowledge to design what goes into a viral clearance study. So you can see that it's much thought and consideration must go into a design of a viral clearance study. So I'm turning it over here to Kathleen so she can get into the details of what you have to do. Kathleen?
All right, thank you, Audrey. I appreciate the intro. Well, good morning or afternoon, everyone. Again, thank you for joining us today. For the remainder of the presentation, I'm going to highlight key considerations in designing your viral clearance study. As Audrey already covered, viral clearance study design for MAB products has been well established over the last several decades. The below tables on this slide, based on the phase of filing, are fairly consistent amongst the industry. For a MAB product grown in CHO cells, phase 1 will typically evaluate 2 viruses, generally an envelope virus like MULV and a small parvovirus such as MVM. In these studies, we'll evaluate 1 or 2 column steps, 1 filtration step, 1 viral inactivation step, such as a low pH or detergent. We design these design these studies to include a process which demonstrates at least 1 robust parvovirus removal step, And I'll go into it a little bit more, but robustness is defined as greater than 4 logs or more log reduction value. Also for a CHO-based product, there are requirements for cumulative clearance plus a safety factor for the retroviruses. When you reach the phase 3 studies, they're going to generally be significantly larger studies. And these will increase the virus panel to include a mixture of large and small viruses, DNA versus RNA viruses, enveloped and non-enveloped viruses, and then further considerations based on your specific product and potential contaminants. In these studies, we'll evaluate multiple columns, your virus filtration step, and your viral inactivation step as well. And then on top of that, you're expected to evaluate additional parameters such as new and aged resin, columns under new normal conditions and those under worst case conditions, carryover, and then any additional considerations based on your actual manufacturing process. In these studies, more samples per process are tested to establish that mass balance, which determines where the virus is actually eluting in your step. When it comes to gene therapy products, the viral risk is considered the same, if not greater, when compared to the recombinant space. The regulatory guidance is fairly heavy on late phase filing, and in those expectations, we see that they're expecting a minimum of 3 virus panel with varying characteristics. You should still distinguish between removal or inactivation of your step. At least one step should effectively clear your non-envelope viruses tested. And then again, you're going to evaluate mass balance in individual fractions. The critical piece of advanced therapy products that Audrey also covered is a thorough risk assessment to justify your study design. Your risk assessment should evaluate the risk based on your helper virus, if it's applicable to your process, cell lines, risk of contaminated cell substrates, adventitious viral contaminants in raw material, and any contamination risk from your operators, equipment, storage, etc. Your study should evaluate steps based on the ability to remove or inactivate virus. If VC cannot reasonably be performed on a given virus for a step, it isn't expected to be performed. For example, here for an AAV study, we wouldn't expect to see nanofiltration for a small parvovirus just based on the filter pore size of the production process. And then, as stated in the guidance, if a helper virus is used in the transfection process, we would evaluate that virus at a minimum. WuXi has performed several viral clearance studies on AAV products thus far for our clients. So on the right, you can see an example study designed for an AAV product that had an adeno helper virus. The virus panel for the study would minimally contain the human adenovirus because it's the helper virus of the process. The remainder of the panel will be based on your manufacturing process, materials, and risk assessment. But it could include viruses such as Rio3, PRV, MULV, and or a virus like SV40 to challenge your viral filtration step. Gene therapy is not as consistent or predictable as in the MAB space, but I do have some expected clearance for steps that we have performed here at WuXi. For detergent inactivation, we still expect fairly robust clearance where we could see greater than 6 logs for your envelope viruses. Affinity capture columns have a range of 2 to 4 logs of viral clearance. Your polishing columns are a little less consistent. Some may have 2 logs of clearance, and some can be robust with greater than 6 logs of clearance as well. Your nanofiltration is still expected to be a robust step, except for obviously we wouldn't test the small parvoviruses there. And then we have seen a variety of heat inactivation, which results vary based on the temperature and hold time. So on this slide, I have a really simple schematic of general purification process. The purpose of the purification process is to contain multiple and orthogonal means to separate and purify product from other cellular and growth media components. Historically, we have seen complete or robust clearance in steps such as chemical inactivation, like your low pH or detergent, the AEX column, and the nanofiltration. The results of other purification steps can vary extensively based on the columns, your resins, your viruses evaluated, etc. If your gene therapy production has less steps for product purification than a typical MAB production, or you have steps that aren't intended to remove or inactivate virus, then your study design will really rely heavily on those process steps that are expected to have robust viral clearance. Once your manufacturing process is designed, we'll move into assessing the appropriate virus panel for your study. The viruses in your study should be based on viruses that may contaminate the product and represent a wide range of physicochemical properties. So the ICH Q5A revision 2 specifies that these viruses fall into three categories. They are relevant viruses, specific model viruses, or non-specific model viruses. Relevant viruses should be used if possible, as they represent viruses that are likely to contaminate the cell substrate or reagents materials used in the production process. A specific model virus may be used if the relevant virus is not available or it's not suitable for a cell-based assay due to the titer or assay suitability. The specific model virus is a virus that is closely related to the known or suspected viral contaminants and has similar properties. But a non-specific model virus may also be used when the purpose of your study is to characterize the capacity of the manufacturing process to remove or inactivate viruses in general. Throughout the ICH Q5A, there are several examples of viruses used within viral clearance studies based on your source material. If a client does not have the panel established, WuXi will help refine the panel by the client, and it is done on a case-by-case basis. That's because we have to factor in all of the variables that contribute to your production process. This slide shows some of the viruses available that can be selected, and again, they have a range of characteristics from size, from small to large, high resistance to low resistance, enveloped and non-enveloped. Ultimately, the choice and number of viruses will be dependent on the production process and the quality and characterization of the cell lines. So now we'll move into the how of viral clearance. Virus reduction will be calculated for each run of each process in the study. The reduction is presented as a logarithmic reduction value, or LRV. We obtain the titer to calculate LRV using one of our key assays. So one example of an assay that we utilize here is the qPCR. With qPCR, we can measure the amount of specific virus DNA or RNA in a given sample. The output is given as genomic copies. This assay is highly specific and highly quantitative. But the drawback is that qPCR measures any DNA or RNA of the virus in a given sample, so it may overestimate a live infectious virus. We also offer the TCID50 assay. This assay is performed in 96 well plates. The assay scores are based on the percentage of cell death with wells scored as positive or negative for viral infection. However, limitations to sensitivity occur at lower concentrations of infected wells. In those situations, alternative calculations are then needed when only a few wells of critical samples are infected. Additionally, in our labs, it's a little bit more difficult to increase the TCID50 assay for large volume testing. And in the next slides, I'll explain the benefits of large volume testing. So WuXi's preferred method when possible to determine virus titer is the plaque assay. The assay is highly sensitive, has higher sensitivity than other infectivity assays. It'll measure relevant infectivity of a given virus within a sample. By placing the sample of virus on the monolayer of cells, plaques will form in the cell monolayer, which indicates cell death, and then they are counted to obtain a virus titer. Most importantly, it is much easier to scale up the test for testing large volumes. So utilizing the power of the plaque assay, large volume testing for steps where we expect full clearance can lead to increased log reduction values. So here's just a pretty picture of our plaque assay. But in this slide, I show a very simplified calculation to demonstrate increased log reduction value with large volume testing. This example assumes a 200 ml load and filtrate, and if that volume sounds very large for you, it can definitely be scaled down based on your process volumes. In this slide, the first volume of the filtrate sample shown is a standard volume tested in our plaque assay. When about 8 logs of virus is in the load sample, we can see 5.3 logs of virus reduction with a standard 1.5 mils of sample tested and assuming no virus was detected. When you increase the volume by simply plating more of your filtrate, the amount of log reduction will also increase, assuming little to no virus is present. So if you go down to the next line, when 21 mils of sample is tested, we now can achieve almost 6.5 logs of clearance. And if we go up to 70 mils, we may see greater than 7 logs of clearance. This is a really simple to execute change, which adds greater than 1.5 logs of virus reduction for a single step without adding additional process steps or samples. Large volume testing for product containing samples could allow a client to evaluate less process steps overall and reduces the study cost and time. So that can be shown here on the next slide. So here at Wuxi, we have developed a fine-tuned approach to the virus spiking strategy using the plaque assay, a targeted viral load, and large volume testing. Instead of the percent spike approach shown on the left, we will spike targeting a specific viral load per run. With this, we spike virus at the quote right concentration, where the virus titer is considered so as to not overload your system, but will still be a sufficient amount of virus per regulatory guidance. On the right, you can see that we obtain increased LRV with Wushi's targeted viral load and large volume testing approach for each step. And most importantly, the results are more reproducible and consistent run over run. This increase in LRV per step could mean that, if you're going to get 6 to 7 logs of clearance in a couple of steps, you may not need steps that only obtain 1 to 2 logs of clearance. And then just real briefly, I'd like to switch goals into a couple more of the important updates from the ICH Q5A revision 2. So they're shown here as prior knowledge and the platform approach. So while prior knowledge and the platform approach have been accepted on a case-by-case basis by regulatory bodies, this new revision specifically addresses these scenarios, which is excellent for the industry. Prior knowledge can be used for well-defined applications. So they even give an example in the guidance that protein A has demonstrated to show similar or greater removal with aged resin. Therefore, it is no longer expected to be evaluated for both new and aged resin in phase 3 studies. Prior knowledge may be applicable to other chromatography steps, but in order to claim those, you should demonstrate that through in-house experience and detailed justification. Additionally, now the guidance allows for use of non-cell-based assays as part of your viral risk assessment. The new revision does a great job detailing where platform approach can be applied for specific steps that have been established to be non-product specific. 3 steps are highlighted in the guidance, but you still must assess your process's critical factors that impact the viral removal or inactivation. So those are explained for the low pH, that would be your pH of your step, your incubation time, and temperature. For detergent or solvent detergent, those include the detergent step, incubation time, temperature, and any prefiltration due to aggregation. And then for filtration, those factors include your volumetric throughput of the intermediate, throughput of the buffer flush, and your pressure. These are further expanded on. It's actually Annex 5, so apologies for the typo. Annex 6 is specifically in regards to AAV products. So, but you can reference that in the guidance document. So with that, I'd like to say thank you for your time today, and I believe we will turn it over for questions.
Yes, absolutely. Thank you both, Kathleen and Audrey, for such a great and insightful presentation. And now we're going to start our Q&A. So I would invite you to join me on camera, Kathleen, please. And to our audience, please keep your questions coming in. We will get to as many as we can today and answer any others via email. And I would also like to draw your attention to the resources tab, which can be found on the right of your screen. So Kathleen, could I please ask you to join me on camera?
Can you see me?
We cannot see you, but it should be fine as long as we can hear your answers, obviously.
I have
But
to.
you can always click on the camera icon at the bottom of your screen. And I think just to kick things off, since we can hear you, we can always start with the first question.
Okay.
Um,
I'm sorry. It says the camera's on.
okay, yeah, that's completely fine.
Okay, good.
So, are gene therapy companies doing a phase 1 study or just waiting for phase 3 market access?
And so that's a great question. So ultimately, the decision to do a phase 1 AAV study is going to be based on your risk assessment. It is not specifically called out in the guidance that you can or cannot surpass it. Ultimately, if you do have known viral contaminant risk, with a helper virus, we would recommend that you perform a viral clearance study. Um, our experience in general is that if you have a step that lends itself to viral clearance, it's best practice to perform a study. Um, But there are ways that a client could justify utilizing the other pillars that Audrey covered with the material characterization and batch release testing as part of their risk assessment. Unfortunately, if they don't do viral clearance, I wouldn't be a part of the conversation with those clients. So I ultimately don't have a ton of experience in what that would look like for a client.
Thank you, Kathleen. That was really insightful and such a comprehensive answer.
Mhm.
And we're actually running slightly out of time, so we have time for one more question.
Oh, no.
Um, what nano filters have you used with AAVs?
So with nanofilters, we see a lot of 35N Planova filters. We do see other larger pore filter sizes in the industry as well, but most of our experience with clients is with the Planova 35N.
Right. Thanks a lot. Actually, thank you both, Kathleen and Audrey, for answering those questions. I know there's definitely more from our audience, but that's all we've got time for today. And any questions we didn't get to, we will reply by email.
Sure.
And this webinar will be available on demand tomorrow, so look out for an email from us with the link. And also, we would like to invite you to take a short survey to provide us with feedback on today's webinar, the link to which can be found in resources on the right-hand side of the screen or in the on-demand email. And again, all that's left is to thank Audrey and
Mhm.
Kathleen once more for a great presentation. And thank you to our audience for listening. And of course, we hope to see you again soon.
Yes. Thank you.